Okay, here we go. All right, so on this side here, this one right here, would it be 5 or 3 prime? This is your 5 prime. This is your 3 prime. All right, with the 1, 2, 3rd carbon with the hydroxyl group. So 5 to 3, which means then that this side, notice it's kind of upside down here. So the phosphate, let's take the phosphate sugar base. All right, so this is the 5 prime end. So we start here 1, 2, 3, 4. Here's the 5th carbon, 5 prime and 3 prime, and that's the anti-parallel. Why is it important for DNA replication to know about this 5 to 3 prime direction? How does that relate to DNA replication? Nathan?
Because DNA replication can happen in one direction.
And what is that direction? That's right. The new DNA molecule, the new strand, grows itself into from the 5 to the 3 prime direction. So therefore, it has to be opposite of the template strand that it is being built from. So I'm going to kind of go through this here real quick. All right. So we have these anti-parallel strands, 5 to 3 prime. So what happens is in DNA, I'm just going through this really fast here. We already know about the base pairings. You have the A's go with T's and C's go with G's. In copying it, we have to separate the DNA strands, and the new strand in the lighter blue has to be copied from the 5 to the 3 prime direction. Remember, I showed you this exact picture that we have multiple origins of replication. So this picture here is showing you a little bit more in depth. So here's one replication bubble. What enzyme helps to make that? Helicase. So helicase untwists and breaks the double bond or the hydrogen bonds, all right, between them. What keeps these guys from coming back together and rehydrogen binding? That's a good question. Starts with an S. Single strand binding proteins. Sorry. It's the only one that doesn't end in ACE. So it's not an enzyme, it just stabilizes it. So here we have those guys here. All right. Notice the reason why I want to kind of go through this animation is because it goes through and shows you more specifically about how DNA replication goes in both ways from this replication bubble. When we talked a lot about it in our notes, in fact, at the end of the hour on Friday, I had drawn this and asked you which one was the leading and lagging strand, and so on. And we look at it from the end of the molecule and so on. But really, most replication occurs in the center of this replication bubble, and replication occurs in both ways. And so we have this helicase that then is on each end at these replication forks, untwisting and breaking the hydrogen bonds in both directions. And then these guys here are the single strand binding proteins that keep them from rehydrogen bonding back together. So here's replication. Here is the daughter strand. So we're looking at just one side. So if we took double-stranded DNA and separate it, we're looking at just one side of it. The same thing would be going on in the other side of it. So looking at this strand of DNA, let me ask you this. With the complementary strand, so we're going to bind and make a second side to this. Would it start from the bottom and go up, or would it start from the top and go down? Don't yell out your answer, just think for a second. Somebody tell me where I would start. What am I looking for to answer this question? Nathan?
Which side is the 5 prime and which side is the 3 prime?
Which side is the 5 prime and which side is the 3 prime? And so when you look at your template strand here, this top is the 5 prime, and it says 5 prime here. You probably can't see that very well, but if it didn't say 5 prime here, how would I know it's the 5 prime? It's the 5th carbon, or it has a phosphate group. If you can just remember the end with the phosphate group is the 5 prime, then you know that the other end has to be the 3' here. So that's your original strand, but remember your growing strand, when you replicate it, that's the strand that has to grow in the 5' to 3' direction. So therefore, if this is the 5' of the template, then the new strand, this is going to be the 3', and this is going to be the 5', and the new strand has to grow from the 5' to 3' direction, and so therefore, if you in your head said it would grow upwards, then you were right, all right? So it would go from the bottom to the top, so the new strand grows in the 5' to 3' direction. What adds the nucleotides? DNA polymerase, remember? 3, yeah, that's it. All right, so in comes DNA polymerase 3. Kind of was cut off as part of the quirkiness of translating it to Google Slides, but here's DNA polymerase 3. First of all, DNA polymerase 3 really, realistically wouldn't work unless what was added first? A primer, right? So we'll just pretend that a primer got added on. Okay, so there's your primer. Now DNA polymerase 3 can add bases in the 5' to 3' direction. So as the phosphate, sugar, and base growing in the 5' to 3' direction. But if you recall, when we talked about adding the actual nucleotides, we're missing something, what? The energy for bonding these. So the enzyme helps to put the nucleotides on, but if you remember that they're not actually added in the form of a nucleotide. It's in the form of nucleoside triphosphate. And so we talked about that supplying energy. So here's your nucleoside triphosphate. Here's your deoxyribose, here's your nitrogenous base, and here's your 3 phosphates. A nucleotide only has 1 phosphate, this has 3. What's the purpose? So that, remember, just like in ATP, when we took a phosphate off, energy is released. And so we can take 1 phosphate off and energy is released, or we can take 2 phosphates off and energy is released, and then we're left with a nucleotide, a phosphate, sugar, and a base. And so that's what happens to add these onto a growing nucleoside strand. So we have ATP, there's GTP where the base is G, we can break it off and get GMP, TTP, break it off, and this is the nucleotide with the T, and CTP, where we have the nucleotide with the C. And so these are your 4 nucleoside triphosphates. So what happens here is, I couldn't get these little energy things to, this is part of the quirkiness to get rid of. So pretend those aren't here. So you have your primer here, and we're going to grow, this is the 3' end, so this is the 5' end, and we're going to grow down. And so DNA polymerase is going to add these nucleoside triphosphates. They BYO energy. All right, and so bring your own energy. All right, so all right, so here's your nucleoside triphosphate, your A that goes with the T, your sugar, and these are your 3 phosphates. We're going to break off the 2 phosphates. That's going to supply energy to hook this to this molecule, and now we have our next nucleotide. The next nucleoside triphosphate with a G is going to come. Here's your 3 phosphates. 2 phosphates come off. Supplies energy to hook this phosphate to this sugar. Next nucleoside triphosphate with a T that goes with this A to come off and provides the energy to put those together, and so on and so forth. And so therefore, 2 things help: the enzyme DNA polymerase 3, but also the form that comes in as a nucleoside triphosphate to provide the energy there. And so, so it grew in the 5' to 3' direction. Now what this picture, again, pretend like these little energy things in the middle are not here. This picture here is going to show you that, but it's going to show you one other thing, and the reason why the molecule grows in the 5' to 3' direction. So if we look at this, this is the 3' end of this one. It doesn't have the phosphate group, it's just the 3'. This, can't see here, it's off here, but this is the 5' here. So 3 to 5. So therefore the new strand, this is going to be the 5 and grow 5 to 3. So it's going to grow down this way. This strand, this is the 5', so this is going to be the 3'. This is going to be the 5', so it's going to grow up. And so for this one here, if it grows, from the top to the bottom. You need the primer, so there's your primer. And here we go, that's what we just looked at. Here's the nucleoside triphosphates, providing energy to hook them together. And these guys can go continuously from the 5' to the 3' direction. Now this is where I was going to show you why is it that the other side has to start down here and move in this direction upwards, why it can't grow in the 3' to 5' direction. So let's pretend like it tried. So here's primase. Primase adds a primer. So there's your primer. So then we're going to add a nucleoside triphosphate with the T. So here, remember here's the 3' because the 3' end and the phosphate group is the 5'. So notice here it adds the nucleoside triphosphate. But remember when we're replicating DNA now we want to hook this phosphate to this sugar here. And so what happens though is when you add the nucleoside triphosphate, the phosphate groups come off here, and this phosphate is looking to bond with something, but there's nothing to bond with here. And these phosphate groups coming off, when it supplies energy, that's not going to help with putting this phosphate together with this sugar. So what you end up with, if you try to grow it in the 3' to 5' direction, is that they can't bond together and therefore it doesn't work. And so it has to go in the 5' to 3' direction. And so there's no energy to bond there. And so therefore we start in the 5' and work our way up. So here we have the nucleoside triphosphate. Now when that phosphates come off, energy is released to hook this phosphate to this sugar. And now we are building a strand here, the same thing here, and so on. Gets to the end, and what happens? We add another primer here and continue to add and therefore provide the energy to hook them together. And so now we have 2 fragments, and so this side is the what strand? Leading or lagging? This is the lagging, so we have 2 Okazaki fragments, but remember that we have to add them together. In comes DNA ligase. Why do you think she put a little water droplet here? Dehydration synthesis, opposite of hydrolysis. So how do we hook 2 molecules together? An OH off of one and H off of another makes the molecule water, and that'll form that bond between there. And so ligase helps to form that by taking off those OH and H. And now we have 2 molecules that are, I'll say. And so this shows you this in just a little bit different way. We have helicase and the single strand binding proteins. Here we have the 5' to 3' direction, so this can continue on. So that is DNA polymerase 3 going in the 5' to 3' direction. Is that going to be leading or lagging? This is going to be leading. It's going to continue and be able to, as this unzips, as helicase unzips, it's going to keep on going, and it's going to be the leading strand. Here we have, it can't go in the 3 to 5, so therefore we have our lagging strand there, and we have Okazaki fragments, and this is Mr. Okazaki up in the corner there. All right. This is the one that I really want to show you here that shows you how the leading and lagging strand changes when you're looking at a replication bubble. So remember that when helicase unwinds and unzips, you have in the center here, origin of replication, and what happens is this is DNA polymerase 3. After the primer is added, it's going to add the nucleotides. But remember that at the replication bubble, replication goes in both directions. And so when you look at this, so if we look at just to the right of this, so here's where we're going to start replication. If we look at just to the right of this, notice here the new strand, this is the 5' to 3' for the new strand. Why? Because if you look at the original strand, this is the 5' end of the original strand, which means the new strand that has to be the 3'. And then this is the 3', which means that it has to be the 5'. So is this top strand going to be lagging or leading going in this direction? Top one. This is going to be leading. It's going to grow continuously in the 5' to 3' direction, and as it continues to unzip, it's just going to keep on going here. So this one here, as it unzips here, so as it starts here with the 5 and makes 1 fragment, starts again, and it'll make many Okazaki fragments, and so this side would be the lagging strand, right? But look at here, when you make that first fragment, when we make this first fragment and you make, it grows in the 5' to 3' direction, is there anything to stop it from just continuing? No. And so what happens is, when we say that replication goes in both directions, going to the right, this is the lagging strand, but going to the left, this side becomes the leading strand, all right? And therefore the top becomes the lagging, so it has to make fragments in this direction. So that's what we see here, to grow in the 5 to 3' direction. And so therefore it continues in both directions, so it's your leading and your lagging. But when we look at leading and lagging here, when we look at to the left, this lagging strand becomes the leading strand going to the left, and the leading strand becomes the lagging strand going to. All right. Yeah, that was important. All right, so therefore it continues growing in both directions, and remember that there are multiple replication bubbles, and so that eventually they'll all meet and you get your completed DNA strand. What's the purpose of having multiple replication bubbles? Efficiency and speed, all right, so we can replicate it much more quickly, all right? Alrighty, and that's what I wanted to talk about here. This one just shows you, just real quickly here, the need for primers and how what comes. So primase adds the primers. So you only need one at the end of a leading strand. You need one at the beginning of a leading strand, one at the beginning of each Okazaki fragment. What comes in and replaces those RNA primers? DNA polymerase 1 will come in and replace it. And so what happens, here's DNA polymerase 1, and look at what the Okazaki fragments. They replace the RNA primer and replace it with DNA, and ligase hooks them together. So in the Okazaki fragments, the primers all get transferred to be DNA nucleotides, and therefore you have one complete DNA strand. The problem here at the end is that you have the RNA primer that's taken away, but it cannot be replaced with DNA. Why? What's the problem? When we had the primer to begin with, think of what was the purpose of having a primer to begin with? To start it. Why do we need a primer to start it? Because what can't start it? Yeah, DNA polymerase 3 cannot start a strand. It can only start, it can only add nucleotides to pre-existing nucleotides. So it's kind of like if you were writing a sentence. It's like you are incapable of starting a sentence. Somebody has to write the first few letters. All right, and then once they get it going, then you can continue on and start it. But DNA polymerase cannot start it. And so therefore, when you take the RNA primer off, we're missing pieces of DNA. And that's where I talked about at the end of the notes that every time you replicate DNA, the ends get shorter and shorter. That's not a really big deal for a while because at the end we have these, Houston, we have a problem. All right, because we're not able to replace the DNA. Yeah, I wish the sound was because she had quacking noises and like all kinds of stuff. Man, it kind of made it a little exciting.
Aren't those ends just like they don't code for anything, they're just bits of...
Yeah, so for a while that's not a big deal because the ends, we have what are called telomeres at the end, which is a repeating segment of DNA that doesn't code for anything. So when we have this missing, it's not a big deal because let's say the genes don't start till right here. So every time this DNA replicates, it gets shorter and shorter and shorter and shorter, and that's not a big deal for a long time. But it says about limit to around 50 cell divisions, about after 50 cell divisions, it becomes short enough where it starts eating into your DNA and into your genes. And remember I said that that's what helps with the aging process. All right? So we have this only in what kind of cells? Only one type of cell in our germ cells. Those are the cells that make gametes. We have this enzyme called telomerase that replaces these ends. And so that's really important because when you pass on your DNA to your kids via your egg or sperm cells, you don't want your kids to have this shortened piece of DNA. You want them to have their full chromosome, and so on. And so this as I said, is believed to be part of the aging process. So there's a lot of research being done about this enzyme and how we could prevent our telomeres from being shortened and therefore delay aging or stop aging. You know, there's a whole host of research being done about that. Because imagine the business. Right now, the anti-aging business is a big deal with even just lotions and Botox and, you know, all kinds of things trying to fix and make people look younger. But if you could, from the cellular inside out, actually slow or stop the aging process, it's a big business.
So, like 50 cell divisions, like how long is that thing? Like,
Depends on the cell and how often they divide, because different cells divide differently and so on. So, yeah. And then,
Isn't it for, I know for some marine animals, they don't have that telomere like shortening, so they have something where they never actually stop growing. They just, you know, like I think the manta ray is one of those animals that like at like 30 years old it can just get huge and it never stops growing because the telomeres never shorten.
yeah.
So they people can get to that maybe.
Yes, and then somebody probably is.
Yeah.
Yeah. Yeah. All right. All right. So that's what I just wanted to just go through that animation and show you how the DNA replicates through this animation. All right. So now we're going to switch gears to chapter 17, which talks about then what is it about DNA that allows us to make proteins, which gives us our traits. So this is why we want our DNA to have, our cells to have all the appropriate amounts of DNA, because DNA codes for making proteins. So we've talked about that DNA codes for making proteins, but we're going to look at the nitty-gritty in terms of how it does that.
show off.
Crazy day today.
Oh my god.
I changed the map.
PowerPoint is different.
You need to chill. You need to just relax.
It is different.
All right. So what we're going to look at is DNA, how the information is coded in the sequence of the nucleotides, the A's, the T's, the C's, and the G's. Oh my, this doesn't erase very well. And so then how does this relate back to genetics? This is terrible. This doesn't want to erase.
Yeah, Sharon had this spiel of how she went to her site as like an experiment because she wanted to prove to her concept of phrase because she would push so hard to erase it and it would.
And this was a brand new marker too. And this is a marker.
Because she would always complain about how it was in her room, like move it all through time. And she's like, yeah, I can see it got better.
All right.
It's only really one or two.
So we're going to relate this back to our appearance. So how do the orders of the A's, T's, C's, and G's give you your phenotype, which is by making proteins. So we're going to look at how DNA, the information there is actually translated into a protein that's going to give you a particular trait in genetics. So before transcription and translation, which are the two major parts of protein synthesis, before those were discovered, there were many questions as to what caused metabolic errors. And so Jared was the first man to suggest that genes code for enzymes that dictate phenotypes of an organism. We call these, these metabolic errors, inborn errors of metabolism. And so, remember DNA is a double-stranded molecule. A gene is a section of that DNA. And so, so he said that there are genes that code for making enzymes, and these enzymes are giving you your trait and therefore your phenotype and your physical appearance and so on. And so eventually they called this the one gene, one enzyme hypothesis. And they studied genes, and we still do study genes based upon when that gene isn't working properly. So for instance, we use genetic disorders where when we research, we say, okay, this person has this genetic disorder because this gene is mutated. So then you can look at and say, okay, this gene makes a particular protein. These people are not making this protein. What symptoms do they have? What's it's affecting? And it can tell you kind of what's working backwards by the gene not working. You can go back and say, okay, what's not happening in them? And it tells you what the gene does. Does it make sense? All right, so working backwards with that. All right, so next comes this guy named Beadle. He went right along with this one gene, one enzyme here, proposed a similar hypothesis. He was studying eye pigment, so in fruit flies, we talked about that, red and white and so on. And as time went on, more biochemists performed different experiments that supported this idea of this one gene, one enzyme hypothesis. So Beadle and Tatum, this is a popular experiment. They experimented with bread mold. Bread mold is hardy. It can grow on minimal medium because it can use various pathways to synthesize everything it needs to survive. Minimal medium is a culture medium that they grow the bread mold on. It contains everything necessary for growth for the wild type. Remember, that's the normal type of bread mold. And so some examples of some things in there: inorganic salts, carbon source, vitamins, water, and so on. So minimal medium because it's the minimum that they need to survive. So then, remember, they're looking into one gene makes one enzyme. And so next what they did is they took this bread mold and they exposed it to X-rays. And X-rays are known to change your DNA. They mutate DNA. So to create a variety of mutants. Most mutants could survive on complete growth medium supplied with all the vitamins and minerals. But then what they did is they looked to see how they responded to other growth mediums, so various growth mediums, those lacking certain nutrients required for growth. And they looked closely at a pathway that says that synthesized arginine. Arginine here, let's put in parentheses, arginine is an amino acid needed for growth. So for the bread mold to grow, it needs this amino acid. So two different ways you can get the amino acid. You can give the bread mold the amino acid. So you can put it in the medium so that they have arginine at its disposal. Second thing is the bread mold can make arginine. So you can make it. Now notice here, so that's why it says that synthesized arginine. So bread mold has the ability to make this molecule. And so they mutated these bread molds, and they found that each mutant was unable to carry out one step in the pathway for synthesizing arginine because it lacked the necessary enzyme. They also noted that the mutants would survive if they were grown on a medium supplied with a compound made after the defective step. So let's go here. Each mutant was unable to carry out one step in the pathway for synthesizing arginine. Pathway for making arginine. Remember when we talked about metabolic pathways? So molecule A gets converted to molecule B, which gets converted to molecule C, which then converts to molecule D. This is a pathway. So this is your beginning. This is really what you want. This is your end product at the end. So in this particular case for bread mold, the end product is arginine. So when it says here that they went through a pathway to make it, they took one molecule and converted it to another, which converted it to another, and then eventually got arginine, which they need to survive. Now, if you remember when we talked about metabolic pathways, each step in the pathway is catalyzed by a different enzyme. So enzyme, let's say 1 here, speeds up this reaction A to B. Another enzyme converts B to C, and another enzyme C to D. And in order to do this reaction, oftentimes it'll say, this is the chemical reaction, this arrow here represents the pathway here. So to get A gets converted to D, you need each one of these enzymes, okay? So, so what they found is this, all right? They hypothesized that they hypothesized about the arginine synthesis pathway. We'll look at it in just a second. They looked at 4 strains of the bread mold, N. crassus, the bread mold. And they found the mutants could only grow when the compound is supplied after the defect. And so this confirms their 1 gene, 1 hypothesis idea. Let me explain what you're looking at in this picture here. So we're going to look at it in columns here. So let's just look at the first column here. Here's your wild type. Wild type is normal bread mold. So this is without exposing it to x-rays. This is just normal bread mold, and they grew it on that minimal medium. So this is a test tube, and this is a solid that has the minimal medium that has all the nutrients and things like that, the minimum that the bread mold needs to survive. The colored part on top of this is supposed to represent the growth of the bread mold. So in this particular case, the wild type normal bread mold grows on this minimal medium. Okay, so that's that. So, so they're looking at the synthesis of arginine. So if this, and the wild type, the bread mold needs arginine to survive. So therefore, this bread mold in here is not given arginine, but they must be able to use something in this minimal medium. They must have molecule A, which they can go through this pathway and make their arginine so that they can survive, because they need arginine to survive. All right, so before I go through the rest of this, let's look at this. Let's look down here. What is the pathway to make arginine? So if we look down here, it says precursor. That would be like molecule A here. So that's the molecule, this precursor. This is a molecule found in the minimal medium. So I'm going to write that here. This is a molecule in the, and I'm just going to put MM for minimal medium, abbreviated up here as MM. If you're not going to remember that, write minimal medium. So this bread mold is able to take something in this minimal medium and convert it to, the first in this process, a molecule called ornithine, which then converts it into a molecule called citrulline, which then they can convert it into arginine. And this is what we want. This is the molecule at the end that we want to keep the bread mold alive. And so, so that's your pathway. Each pathway is, each step in the pathway is catalyzed by a different enzyme. So enzyme A, B, and C. And so what they were hypothesizing is that each of these enzymes were made by a different gene. So gene A made enzyme A, gene B made enzyme B, and gene C made enzyme C. All right, and so therefore, because this bread mold on the minimal medium was able to grow, what they said is that their genes must be intact. They must make all 3 of these enzymes and be able to do this whole pathway and make arginine and be able to survive. All right, and so that's what happened. And so there's nothing wrong with these genes. And so then they added, they did grew it on another medium, which is the minimal medium plus ornithine, which is the second step here. They're able to grow minimal medium, and then they added citrulline, and they're able to grow in minimal medium and arginine, and they're able to grow. And that makes sense, because if you're given what they needed arginine. So in this case here, they don't even have to make it, because they're given it and they're able to grow. And that's the wild type. All right, so that's kind of like your control. So then what they did is remember it said that they exposed x-rays, they beamed x-rays at this bread mold. So therefore, when they looked at what mutants resulted, they classified them into 3 categories based upon which one of these mediums that they grew on. So these guys were exposed, these are the ones that are exposed to the x-rays. So if we look at this, here, class 1, they found a bunch of bread mold that grew in this pattern here, where they didn't grow in the minimal medium, but did grow in all 3 of the other cases. So then going back to this pathway here of what happened, they didn't grow on the minimal medium, but if you give them ornithine, they can grow. So basically the fact that they didn't grow on the minimal medium, the fact that they didn't grow on the minimal medium means that they couldn't take that precursor and convert it to arginine like the wild type did. So therefore they didn't grow there. But look at here, if you give them ornithine, if you bypass that first step, if you give them ornithine, then they're able to, they grew, which means that they're able to synthesize arginine. So therefore, this bread mold here has enzyme B and C, but do they have an enzyme A that's working? No. Enzyme A is not working. So what does that mean? That means that when they expose this bread mold to the X-rays, it must have mutated gene A, which then caused it not to make enzyme A, and it can't do that first pathway. But as long as you give it the molecule after that pathway, it can survive, okay? And by the same token, then they found another class of mutants where they didn't grow on the minimal medium or the one with ornithine, but grew on the other two. So therefore, in this case here, they couldn't take the precursor and make it to the end. They couldn't do ornithine and make it to the end, but if you give them citrulline, then they are able to convert citrulline to arginine. All right, so therefore we have a mutation in gene B here. And then lastly, this last one here only grew given arginine, and so therefore could grow. It couldn't even grow if you give it citrulline, so that means it didn't have gene C and therefore couldn't make arginine. But if you give them arginine, they are able to survive. And so this helped to prove this one gene, one enzyme hypothesis, that one gene makes one particular enzyme. So somehow the gene that converts one product to the next in the pathway must have been affected by the X-rays, so different mutations. So one gene must be responsible for one enzyme, which was the original hypothesis. You want to go too fast? One gene must be responsible for one enzyme. We now know, as we know more about proteins, that not all proteins are enzymes. It was determined that not all proteins are enzymes. They are made, all proteins are made by genes though. So the hypothesis was revised. It's not one gene, one enzyme anymore. It's the one gene, one polypeptide hypothesis, because many enzymes are comprised of multiple, I'm going to have you change from proteins to polypeptides. And even this technically is wrong because some genes actually code for RNA, making certain RNA molecules, and that may not become proteins. So not every gene actually even makes a polypeptide. Let me talk a minute about this. Many enzymes are comprised of multiple proteins. So an enzyme, remember when we talked about proteins in the quaternary structure, that one protein can be made out of many polypeptide chains. That's the quaternary structure. Remember that? Hemoglobin was an example of that, made out of 4 polypeptide chains. And the whole thing, this is one enzyme, let's say, and this is its particular shape, but it has that shape because of the 4 polypeptides it makes. And so what happens to get these 4 polypeptides is from your DNA, you have different genes. So you have gene, let's say gene 1 here, that codes for making polypeptide 1. And then here you have gene 2 that codes for making polypeptide 2, and so on and so forth. Each one of these, once this polypeptide is made, it's not a finished protein. It's not until these polypeptides come together to build one particular protein that you have a finished protein. All right? And so that's what that means here. They're comprised of multiple proteins. And not every protein is an enzyme. We have proteins that are pigments that give us our hair color, our skin color, and so on. And those aren't enzymes. So not every single protein is an enzyme. All right? So now we're going to look at then how is it, we're going to start looking at how is it that we make a polypeptide from a particular gene. And what bridges that gap is RNA. So RNA is the single-stranded compound that carries the message from the DNA to the ribosome for translation into protein. So one of the differences between DNA and RNA, recall that DNA has the bases A's, T's, C's, and G's as part of their nucleotide, and RNA is A, U, C, and G. So RNA does not have T's, they have U's instead. And so the order of these bases is what carries the code for the protein, which then is constructed from any or all of the 20 amino acids. And so it's the order of those bases. So this is one difference between DNA and RNA, is the types of bases. So RNA has U and DNA has T. What are some of the other differences between DNA and RNA that we talked about before?
DNA is made out of the sugar deoxyribose and RNA is just ribose.
That's right. So in the nucleotides, DNA has deoxyribose, RNA has ribose. What's another difference?
RNA used the nucleus, or is that mRNA?
Is
Or the-
used in the nucleus?
Well, sorry, leaves the,
Leaves. Oh.
yeah, leaves the nucleus.
Yes, RNA is able to leave the nucleus, DNA is stuck inside. All right, so DNA is in the nucleus. Anybody? There's one other thing.
Is RNA not a double helix?
It's not a double helix and it's not double-stranded either. All right, so RNA is single-stranded and DNA is double-stranded. So that's the other major one. All right, good memory. All right, so let's look at this. There are two major steps to protein synthesis. So going from a gene to a protein, first is transcription, the synthesis of mRNA using DNA as the template. This is similar to DNA synthesis. We'll look at this more specifically. So mRNA is the message from the gene. The M stands for message. Translation is the second step. It's the process that occurs when the mRNA reaches the ribosome and protein synthesis occurs. So your next picture is a picture I've shown you before. When we did cell parts and so on, when we talked about ribosomes and so on. So here we're going to look at just the left side here first. You have a prokaryotic and a eukaryotic cell. So let's look at the prokaryotic cell. Prokaryotic cells do not have a nucleus, so everything is going on in the cytoplasm. The DNA is hanging out in the cytoplasm. And so in protein synthesis, DNA, the instructions to make the protein, is copied onto an mRNA molecule. So now the mRNA has the message. That process is called transcription. So it's transcribing or copying the message from the DNA. Then the mRNA takes that message to the ribosome, that's this brown here, and the mRNA is quote unquote read and tells the cell what amino acid to hook to what amino acid. So each one of these circles here is supposed to represent one amino acid, and it's making a polypeptide chain, and that's called translation. So at the ribosome, you're translating that message from the mRNA and building your polypeptide chain. All of that here is not in the nucleus because prokaryotic cells do not have a nucleus. If we look at the eukaryotic cell, we have our nucleus, which, as Liam pointed out, that DNA stays in the nucleus, mRNA can leave the nucleus. So therefore, mRNA in eukaryotic cells is really important to take that to get that information out of the nucleus. It goes to the nuclear pores to the ribosome. So transcription occurs, so the DNA, the message is copied onto, but notice here we have pre-mRNA. So notice here, we have another step in our cells versus bacteria cells. It's called RNA processing. If we look here from this to this picture, pre-mRNA to mRNA, what happens to the length of it? It shortens. So part of that, we'll look at why, but we cut out part of the mRNA. And then, if you notice the ends, there's this, it's not going to show up, it's yellow on yours, but there's this yellow. They call it a cap and a tail. It helps so that the mRNA, when it leaves and goes out to the big bad cytoplasm, it's not broken up and degraded by enzymes. All right. And then it goes to the ribosome, and the same thing happens translation where it hooks the amino acids together. All right. So we will continue and look at this the rest tomorrow. Maybe. We'll see how the curriculum day